Hi everyone! In this unit, we're going to be discussing the intertwined processes of sensation and perception. Sensation and perception are distinct processes, and it's important for you to understand the differences between the two. Sensation is the act of getting external stimulus energy into the body and turning it into internal neural energy. Sensation, for the image here, for instance, is the light being emitted from your screen, passing through the eye and striking the rods and cones and your retina. Perception is the higher order process of making sense of all of the information acquired by those photons striking the retina. Your sensation system has no problem with this image. It's your perception, or trying to make sense of the image, that makes this such a challenging image to look at in some sense. We're going to begin by discussing sensation and then move on to discussing perception. The complicated and really amazing process of sensation is mediated by our sense organs, the parts of our body that transmute external energy into internal energy. Each of these sense organs have specialized cells in them. We talked in Chapter 2 about how neurons respond to the excitation or inhibition of other neurons. Each of these sense organs contains cells that respond to specific types of external energy. For instance, the eye contains rods and cones, cells that respond to photons striking them. The nose, for instance, contains chemical receptors. The ear contains cilia, or hair cells that respond to vibrations or changes in pressure. The tongue and mouth and upper throat contain a variety of different kinds of taste buds. Those taste buds have different kinds of chemical receptors. Those chemical receptors transmute your food into specific sensations of flavors. And finally, the skin itself contains different kinds of sensory receptors, pressure and temperature neurons that respond to different kinds of energy to elicit these types of sensations. As I've indicated already, these senses are all about detecting and processing external energy. Understanding the world around us is really the process of getting outside energy in. One of the ways that we understand how these organs work is to investigate their thresholds. In psychology, when we're talking about sensation and perception thresholds, what we're talking about are sort of the bare minimum of perceptual thresholds. That is, how often do we notice consciously that we're seeing something or hearing something or feeling something or tasting something? We define the absolute threshold, and I recognize this is a little silly, but we define the absolute threshold as the stimulus intensity that is detected 50% of the time. That is, if I show you a light of a certain intensity, a certain number of lumens, the light that you can detect 50% of the time would be the absolute threshold of your vision. Interestingly, the absolute threshold for light is very low for humans, though not as low as for some other animals. The absolute threshold for human vision represents roughly a candle at 30 miles on a clear, dark night. But if we zoom in a little bit on that curve, what you'll see is that there are stimuli that are detected less than 50% of the time, but are sometimes detected. And if they're sometimes detected, that means they're being sensed. This area, this gap between sensation and perception, are what we call subliminal stimuli. Subliminal stimuli, that literally means below the Lyman, which is the German word for limit, so below the limit. These subliminal stimuli can still have an influence on our higher order cognitive processes. The book talks a little bit about subliminal processing, and they do so in kind of a general way. And since it's an area that I'm particularly interested in, I wanted to take a moment to talk about a couple of things that we know about how the subliminal presence of stimuli can influence our attitudes and behavior. Recently, Barge and Chartrand have demonstrated a few really interesting social perceptual phenomenon that happen when people are primed or when they're given stimuli that affect them even above conscious awareness, but in a very subtle way. Let me give you an example. In one study, these researchers found that participants who were asked to hold a hot coffee compared to those who were asked to hold an iced coffee rated the person that they were interacting with as more psychologically and socially warm. That is, people who were asked to hold a warm coffee, who were holding a warm beverage, transferred that sense of physical warmth into the experience of psychological warmth or pleasantness from the person that they were interacting with. Or vice versa, those that held the iced coffee transferred that physiological sense of coldness into social coldness or distance from the researcher. In a related study, these researchers exposed some participants to a bunch of words related to being old. They also exposed another group of participants to normal words like shirt and butter and iron and things that were of no particular category. What they found is that when the participants left the room, those who were exposed to the words that were related to being old walked more slowly down the hallway, giving some indication that the idea, even though it was outside of their conscious awareness that it might be influencing them, was influencing their physical behavior. Let me give you one more example about how perceptual stimuli can influence our attitudes and our thoughts and our behavior. I want you to say these words with me as they appear on the screen. Soak, folk, poke. And let's repeat them a few times. Soak, folk, poke. Soak, folk, poke. Soak, folk, poke. Now, what's the white part of the egg called? The word that probably sprung to your mind is yolk, which of course is the yellow part. What we've done in this case is activated the neural networks associated with that particular sound, making it more likely that you have the wrong answer than the right answer. This is just to demonstrate how things that are outside of our conscious awareness can influence our attitudes and behavior quite strongly. In order to understand the sensation systems in particular, we're going to take a very close look at the human vision system as an example for the other sensation systems. The reason for this is that the vision system has been studied more extensively than the other systems to some extent, and it's much more complex. So if you understand vision, you can understand the other systems clearly. In order to understand any of the sensation systems, we have to understand the energy that's coming into that system. In the case of the stimulus energy for vision, that energy is light. For the purposes of understanding vision, we should think about light as a wave. And as with any wave that you might have learned about in a basic physics course, that wave has both a wavelength and an amplitude. The wavelength is the distance between peaks or valleys of the same wave, and the amplitude is the distance between a single peak and a single valley. That is, how high versus how low does the wave go. In the case of light, the wavelength, or the distance from peak to peak, determines the hue, or the color that we perceive of that light. Shorter wavelengths represent bluer colors, whereas longer wavelengths represent redder colors. But the amplitudes of the wave also matter. Higher amplitude waves are more intense and are perceived as brighter. Lower amplitude waves are less intense and are perceived as more dim. These waves of light really represent only a very small section of the electromagnetic spectrum. The wavelengths that the human eye can sense are from just below 400 and just above 700 nanometers. What many people don't realize is that this electromagnetic spectrum, the light that you perceive, is the same energy at different wavelengths that we use to broadcast radio. Next, we're going to talk about the anatomy of the eye and what happens to that wave of light as it strikes the surface of the sense organ. As light enters, the first structure it encounters is the cornea. This cornea is a clear coating on the surface of the eye that protects the sense organ from damage. The cornea also bends light so that it will strike the lens at the appropriate angle. Behind the cornea, light passes through the aqueous humor and through the pupil, a hole in the opening of the iris, the colored part of your eye. Just behind that hole, the pupil, sets the lens. The lens is a relatively rigid structure, stretched by some of the strongest muscles in the body. The lens changes shape based on what you're attempting to focus on, so that the object of your focus is directed at the best part of the retina. This now focused light passes through the chamber of the eye and the vitreous humor fluid, and then strikes the retina, which we'll zoom in on here. The retina is positioned at the back of the eye. Interestingly, light striking the retina must first pass through the nerve fibers and other intercommunicative cells before it strikes the photoreceptors. The photoreceptors are the cells that respond specifically to the light energy of a photon. Those photoreceptors are rods and cones. When a wavelength of light strikes a photoreceptor, it triggers a reaction through the chemical rhodopsin that encourages that cell to fire an action potential. That action potential stimulates a chain reaction that ultimately results in the perception of that photon, or the seeing of that candle at 30 miles. These ganglion and bipolar cells serve an important function in helping to inhibit and control and moderate the responses of the photoreceptors in the cases where you're receiving constant stimulus energy. In vision, you're constantly perceiving light, and that light energy is changing all around you. The ganglion and bipolar cells help to manage that energy. But they can also be tricked. Let me show you a quick example that you might be familiar with. The reason why a curveball appears to curve. So first, take a close look at this image and follow the ball as it spins on the way down. Follow it from the top of the image all the way to the bottom. On its next pass, what I want you to do is take a look at the blue dot and watch the spinning ball from out of your periphery. What you should notice is that the ball appears to drift from right to left at about a 20-degree angle. Now, as the ball passes this time, shift your perception back and forth between the spinning ball and the blue dot on the right. The ball should appear to jump suddenly to the left. For any of you that have played baseball and have attempted to hit a curveball, this is what happens when you suddenly shift your focus from the pitcher to attempting to follow the ball. It appears to jump to the left and makes this particular spinning pitch really difficult to hit. The reason this happens is a shift from foveal to peripheral viewing. As you can imagine, light energy passing through all of those cells before reaching the photoreceptors might distort your vision. That's why the cornea and lens work together to focus light and focus your attention on the fovea. The fovea is a part of the retina where all of that other mass, the ganglion cells and bipolar cells that help communicate between the photoreceptors and the optic nerve, are pulled aside, and you have the most density of cones, which are sort of our high-definition color vision photoreceptors. This allows the object of our focus to sort of have direct access to the photoreceptors, making our vision most accurate on exactly what we're focusing on, and sort of blurring vision at our periphery. Now that that stimulus energy has been transmuted from electromagnetic into neural, that stimulus energy travels up the optic nerve, across the optic chiasm, and directly to the thalamus, like we talked about in Chapter 2. The optic chiasm is where different parts of the eyes, and thus different parts of the visual field, cross over to the contralateral or opposite side of the brain. The reason that this is particularly interesting is it means that the right visual field is represented in the left side of the brain. That is, anything that you see to the right of your vision is represented in the left side of your brain, and vice versa. Anything you see on the left side is represented in the right side of your brain. Remember that video I asked you to watch for Chapter 2, the split brain study? When those two sides of the brain are unable to communicate with each other, only the right visual field, which goes to the left side of the brain, is able to access the verbal component of the brain. Once that information leaves the thalamus, it goes back to the occipital lobe. And you'll remember in Chapter 2 how we talked about the occipital lobe is primarily made up of all the different processes that are necessary to perceive the complexity of our visual world. One such component of vision is feature analysis. Feature analysis is how we understand the orientation or shape of an object. What we understand from studies of other animals is that there are neurons that specifically fire when they're presented with a specific orientation. They fire in a different pattern. So that is, there are neurons in your occipital lobe that will fire very excitedly to a 15-degree angle, and that will fire a little less to a 20-degree angle, and will fire not at all to a 30 or 45-degree angle. Just like our bipolar cells, we can also trick our feature analysis sector of the brain by exposing it to the same stimulus for an extended period of time. Let me show you an example. What I'd like you to do is to make this image full screen and to stare at this image for the next 30 seconds. Okay, now look away at a flat surface. What you should see is that surface undulating and writhing. That's because your feature orientation cells have developed a pattern of firing, and it's taking them a few seconds to adjust that pattern of firing back to normal. Okay, I hope your eyes have resettled. Now, there are all sorts of different types of these feature analytic sections of the brain. For example, I'm not going to spend time in this video discussing the opponent process and the trichromatic theory of color vision, although it perhaps is the most important piece of the occipital lobe's visual processing. Study that section of the textbook closely. Now we're going to move from the process of sensation, the process of transmuting external energy into internal energy, to the process of perception, the process of piecing all of that neural imagery back together to represent the world around us. When you think about it, what we've done is broken down the complexity of the world around us into these tiny little nerve impulses that are only responding to very specific parts of the world around us. When your occipital lobe, and particularly when your parietal lobe, gets the input from all of these different areas of cortex, what it's being told is left-oriented 15 degrees, kind of bluish, right in front of us with a shadow. It's getting all of that information all at once, and the brain has to make sense of that somehow to put together the three-dimensional world that you navigate. The brain does this through a variety of Gestalt principles. I won't get into the details on each of these principles. You should study them carefully in your text. But what I will mention is that the brain is very good at grouping things together. We group things by their proximity, by their similarity, by their continuity, and by their connectedness. So instead of, in the continuity image here, for instance, viewing this as several line segments that occasionally cross, we view it as two intersecting lines. It makes the processing much quicker to be able to group stimuli together. The principles of grouping allow us to put together the stimuli that we're sensing into a perception that makes sense. But sometimes those principles can lead us astray. For instance, we didn't see the gaps in the image below because we were piecing them together from behind what was covering them up. One of the most interesting things to me is how humans are able to navigate and perceive the complexity of a three-dimensional world with only these two little eyes on the front of our face. Depth perception is incredibly important. If we weren't able to perceive depth, as you can see in the images below and how we test when depth perception develops, we might just wander off a cliff without any notice. Let me give you an example. This image was just taken by a colleague of mine in the Vatican in Rome. Look at this image and try to perceive what's in front and what's behind. How are you figuring out what's in front and what's behind in this image? There are a few different ways that you can do it. The first is what's called linear perspective. That is, the things in the image appear to converge at a point at the end. But since this isn't a particularly long shot, you can't really see how things are converging at a point in the middle. So linear perspective doesn't provide you a lot of information here. The first and most obvious example is interposition or occlusion. The things in front seem to occlude or shadow or shade the things behind. For example, the light coming through the window in the back is occluded by the throne in front. Another is relative size. The things in the front of the image, for instance the column on the right, appear to be much larger than the things in the back of the image, for instance the column in the very center. Another clue you might get from this image is the texture gradient. Things in the front of the image appear much more rough and detailed than things in the back of the image, which appear more blurred and uniform. What you might notice if you're paying careful attention to your textbook is that all of the things I demonstrated here are monocular clues. Why should this just be monocular clues when you're looking at it with two eyes? Obviously this was taken with a single lens camera, so it only had one eye. But if we had two, if you were standing in this space, you would also have the binocular clues available to you to determine depth, and these are particularly powerful. One of those binocular cues is convergence. That is, how far do your two eyes have to look towards the center in order to see something? The closer to the center, the closer an object is perceived to be. The other is retinal disparity. How different, how far apart are the images on the two retinas? The farther apart the images are, the closer that object is. Depth perception is very difficult. Some animals do it better than others, and humans are very good at it, especially given their physiological limitations of the placement of their eyes. Some of my very favorite things about the sensation and perception chapter is that we get to talk a little bit about perceptual illusions. What you'll find in this class is that we often understand how the human brain works when it's going right by looking at how the human brain goes wrong, when it makes mistakes. We talked about this already and a need for psychological science because we make heuristic mistakes when we misjudge the probability of things. In the case of perception, we can understand a lot about how the brain perceives the things it senses by seeing when perception goes wrong, when we perceive perceptual illusions. That is, when we're tricked by something that isn't really there. Instead of showing you a list of my favorite perceptual illusions, though you've already seen the curveball and the motion after effects, what I'd rather do is ask you all to share them. I'm going to create a new post in the class-wide discussion forum asking you to post your favorite perceptual illusions. Look around on the internet, search for perceptual illusions, and see what you can find. Describe what kinds of processes in the brain might be going on that provide that particular illusion.